So I will just dive right into this intro to astrobiology. My name is Dr. Sarah Rugheimer. I'm a Hugh Price Fellow at Jesus College and a Gladstone Research Fellow at Oxford. And I am interested in astrobiology, which is how to detect life on other planets, as well as looking at the origin of life on Earth. So I'll be talking a bit about the whole field of astrobiology to give you an idea of what types of majors and careers you might be interested in if you're interested in this field to go into. It can basically, you can come at it from any angle, from chemistry, from biology, from astronomy, from geology. And so I'll be hitting a little bit on these as well as my own personal research area. And I have a few parts saved for career, sort of general career thoughts, since I came from a rural background in Montana. And I'll just kind of talk about how I got here. Okay, so I'm interested in this question of, are we alone in the universe? And you might have heard of SETI, that's the search for extraterrestrial intelligence. And that's been looking for signs with radio signals, looking at the cosmos to see if there's any intelligent civilizations beaming radio signals. We've been looking for 60 years, and we've not seen anything. We've not seen a hello Earthlings sort of call from the cosmos. We've mostly been just met with silence. And so my question is, are there aliens? And I'm not really talking about these sorts of movie aliens. When I think about aliens, I'm really thinking about microbes. So single-celled microorganisms living on another planet. Because that's actually what dominates our own Earth's atmosphere, is microbial life. And indeed, our own Earth's biosphere, most of it is microbial. So I'm going to read this quote. It's rather long, but I think it's beautiful, from Carl Sagan. And you can look at this image over here to the right, and it's this little pale blue dot that's been magnified for you in the cutout, and that's Earth, from about 6 billion kilometers. It's out by the orbit of Pluto. That Voyager, when it went out there, it turned back and took this image of Earth, and this is Earth as it would kind of look like to an alien astronomer, right? It's just a single pixel of light. So this is what Carl Sagan wrote about that. Consider again that dot. That's here, that's home, that's us. On it, everyone you love, everyone you know, everyone you've ever heard of. Every human being who ever was lived out their lives. The aggregate of our joy and sufferings, thousands of confident religions, ideologies, and economic doctrines. Every hunter and forager, every hero and coward, every creator and destroyer of civilization, every king and peasant, every young couple in love, every mother and father, hopeful child, inventor or explorer, every teacher of morals, every corrupt politician, every superstar, every supreme leader, every saint and sinner in the history of our species lived there on a mote of dust suspended in a sunbeam. Our posturings, our imagined self-importance, the delusion that we have some privileged position in the universe are challenged by this point of pale light. So this is sort of what I want you to start thinking about, is from astrobiology, we can really zoom out and look at the universe and life in a very broad context. So if aliens were looking at Earth, they would look at that pale point of light, and they would actually be able to break it up into different wavelengths. So that's what you see happening here. You see a spectra, so that's that squiggly line there. And because molecules absorb at very specific frequencies, they leave a fingerprint into that light. And so we can actually see signs of oxygen and ozone, or methane, carbon dioxide, vegetation even. And so if an alien astronomer were looking at Earth, they would see this is the spectrum of Earth, and they might be able to infer that there's life here. And so we've been finding other planetary systems since 1995, so that's planets orbiting other stars, or exoplanets. And, there are just billions of them. And there's maybe 40 billion Earth-like planets in our own Milky Way galaxy. So this is the highest resolution image we have of our Milky Way galaxy. So you can see we're in a spiral arm galaxy, but because we're inside of it, we can't take a picture from above, so we can just see the disk around us. Those two Little satellite galaxies are the large and small Magellanic Clouds. You can only see those from the southern hemisphere if you've ever been down that way. We can't see them from the northern hemisphere. But this is, there's about 100 billion stars in our galaxy, but we've imaged directly about 1 billion of them. And so that's what's shown in this picture. And there are just, hundreds of billions of galaxies. So I'm not sure if you've ever come across this image, but it's called the Hubble Ultra Deep Field. And I'd really recommend, if you just write down in your notepads or wherever you have, you do a YouTube search for Hubble Ultra Deep Field in 3D. It's a 4-minute video. It's beautiful, and it really describes this image in depth. But basically, I'll go through the short story here. So this image that you see is a picture deep into the cosmos. So what Hubble did is it looked at a dark patch of night sky. A patch of sky that's only about the size of a grain of sand held at arm's length. So I don't know if you can see my finger because I can't see myself in the video. But a very small patch of sky on the night sky. And it stared at it, and we didn't know of anything there. And so it stared at it for 10 days the first time, and then 11.5 days the second time. And Hubble is an expensive telescope to have built. It's lasted for decades. It's still in operation. But basically, Hubble is about $1 a second. So this image took 1 million seconds. So it's $1 million image here. And what's beautiful about this image is it looks like just a bunch of points of light. But each of those tiny points of light, if you look at the image closely, each fleck, even the smallest fleck you can see, that's not a star. That's an entire galaxy of hundreds of billions of stars. And you can see some of those kind of zoomed up here in the panel to the left. You can tell that there are galaxies. But even all those little other small flecks of stars, of points of light, are galaxies. It's just the ones that look like galaxies are closer to us, and the ones that are just points of light are further away from us. And so each of those points of light contains hundreds of billions of stars, each with hundreds of billions of planets in those galaxies. So this is the image close up. I'm just going to leave it for a second for you to just kind of contemplate, because this is one tiny patch of sky. And so that we did it again in another tiny patch of sky and saw the same thing. About 10,000, there's 10,000 galaxies in this image that you can see. So if you kind of imagine there being in every little sand grain size of patch on the night sky all around you, 360 degrees, in each one of those is 10,000 galaxies, and each galaxy containing hundreds of billions of stars. And that starts to give you kind of a sense of how big the universe is, which is just utterly mind-blowing, or astronomical, as the term, might accurately be. And space is really big. When we think of just our moon, our moon actually is fairly close, right? And so often we have this conception of something like this, there's the Earth, there's the moon. This is not to scale. If we actually were to draw this to scale, this is to scale. So you have the Earth on the left, the moon on the far right there, and all of the planets in our solar system could fit between the Earth and the moon if we were to lay them end on end. So space is really big. Even the closest thing that we can get to, and we have been to and sent astronauts to, is still very, very far away. So I would also recommend jotting this down and Googling "if the moon were only 1 pixel, a tediously accurate scale model of the solar system." It'll take you about 10 minutes to go through, and there's little fun facts along the way, but it shows to scale just our solar system if the moon is using the moon as 1 pixel, and then all of the distances and size of the sun and everything are scaled to that. And this is, keep in mind, this is just our solar system. The nearest star system away from us is, it's 4 light years, and our solar system is very, we've only sent something 1 light day away. So our solar system is relatively compact and small, and even the nearest star to us is immensely far. So when we think about how to find life, though, there's a few ways that I would like you to think about maybe how we start thinking of the framing of it. One is we want to find exoplanets and solar systems that are conducive for life. And what does that mean? So usually we think of it needing a surface. And why is that? So if you're in a gaseous planet, it's hard to have chemistry happen, for chemical reactions to occur, for life to interact with its environment in a purely gaseous environment. And so we often say that the planet has to be rocky or has a surface, and it has to have liquid water. Now this is for life as we know it. And I'll be talking about life as we don't know it later, and other solvents that might not be liquid water. But on Earth, every single form of life that we've ever come across uses water in a fundamental way. And water is abundant everywhere we look. So this is one of the reasons why we're searching for places with water first. So this leads us to the concept of the habitable zone. So this is where around different stars, because stars can be smaller or bigger. And if they're bigger, then the habitable zone's further out, and they're smaller, they're cooler, so the habitable zone's closer in. So this is where there can be liquid water stable on the surface of a planet. Now that's not to say other environments are not habitable within the solar system. For example, we think that Europa is a habitable environment. That's a moon of Jupiter, and that has a liquid water ocean under an ice layer, but it doesn't have an atmosphere that we could look at from a distance. So the habitable zone is really a concept for astronomers and how to see life from these interstellar distances, from another star system. Within our own solar system, we can search for other places for life because we can actually send robots, landers, things to explore it. But Europa as an exoplanet doesn't have an atmosphere, and we couldn't see it, because that's how we can actually determine what's in these planets, is by looking for things in the atmosphere. So we've actually found about 55 habitable planets so far, and those are relatively close by, given the scale of the universe, and that's not very many, but it's more than one, right? So we've been finding these planets which could be habitable. Now that doesn't mean they are habitable. This just means that theoretically there could exist a condition that allows liquid water to be on the surface of those planets. And so that's where we're going to look first when we do observations of the atmospheres of planets. We'll be looking, at these habitable planets in start when we do that, as well as other types of planets, to see just what's out there. And I want to make sure to just reiterate this point, that just because a planet is in the habitable zone doesn't mean it's habitable. There are so many factors that go into whether a planet may or may not have life that we don't really even understand yet, and we're trying to learn more. And each of these things here is a whole discipline, a whole research area within astronomy, planetary physics, geology, stellar astronomy. And so all of these could play a role, and we don't really know. So there's just different, all of these are different research areas that could link to astrobiology, and the planet that might be habitable it might not actually have life. So just because a planet is habitable doesn't mean it has life, for example. You might have heard of the TRAPPIST system in the news. It's been in the news the last few years. It's a system of 7 Earth-sized planets. Now, just because they're Earth-sized, it doesn't mean they're like Earth. But what's really exciting about this is the star is really dim. It's a really dim star, very cool. So all the planets are super close in to the star. It's much smaller, more compact. I think, in fact, all the planets are within our orbit of Mercury, which is the closest planet in our own solar system to the sun. It's a stacked system of these 7 planets. And what's cool about it is there's 3 of them that are in the habitable zone. And this is an artist's image of standing on one of those habitable planets. And those other 2 planets you can see in the night sky, those would actually be the size of our moon in our own night sky. So you could actually see continents and oceans, perhaps, on those other planets with your naked eye, if all of these 3 habitable planets are habitable. And so I think that's a really cool thing to think about. So there's been a lot of press around this, and sort of, could you planet hop between these planets because they're relatively close? And so this is the TRAPPIST system of the planets. You can Google and find out more about the system. It's a very exciting system compared to the sizes of those planets compared to our own solar system rocky planets. And you can see that a lot of them are around the size of Earth and Venus. And then, like I said, 3 of them are habitable, which is very exciting. So in our own system, we have Venus, which is on the inner edge of the habitable zone, maybe was habitable at some point in its past. And then you have Mars, it's on the outer edge of the habitable zone. Maybe it has life in the subsurface, but neither Venus or Mars have something that's clearly life when you just look at it, because, in the same way that Earth does, with lush trees and continents and oceans and kangaroos and all of that. So if there are life on either Venus or Mars, it would be microbial life, very in a limited niche environment that may be stemming from when it had more habitable environments earlier. But in the TRAPPIST system, you actually have 3 solid habitable planets, which is exciting. And so we don't know yet whether whether Venus and Mars have life. There's a lot of debate. You'll hear about this debate as you read more astrobiology news or hear in the news. And we're searching, because these are planets close to us and they at once had habitable conditions. And so we want to see, what's the frequency of planets that have life? And even just answering this question of what is life? How robust is the discovery? Should you believe first announcements of discoveries? This is we could spend the whole discussion on this, because I think it's actually really interesting, is how do we vet claims? How do we know something's true? And what is the burden of evidence required to say that something's life on Venus or on Mars? And so I want you to think about that, because I might, if we have time, that'll be part of the discussion at the end, will be, how do you know? What would qualify as evidence? And why is it harder for these sorts of environments than say on Earth? So going to liquid water, when you can think about what does life need, it needs food and water. So food can come from energy, solar, chemical, it needs something to eat, and then it needs water or another solvent to do chemistry. That's the main reason for it to move around molecules. And by moving around these molecules and electrons, it's able to get energy to do its cellular functions. So when we think about what does Earth life produce, this is the spectrum of the sun on the top and then Earth on the bottom. And the Earth spectrum is kind of 2 humps, you can see. So there's the reflected light, which is the first hump, and the second hump is the thermal emission, so like the heat radiating off of it. And in both of those parts of the spectrum, you have these absorption features of molecules. And a lot of them are produced by life. And this is what an alien astronomer would try to do, seeing Earth, or what we're trying to do when we look out. We want to get lines like this for other planets to see what's in their atmosphere, to see if we can tell if there's life. Now, life is really weird, though, and I just want to take a brief pause to highlight some discoveries which are incredible. So a few years ago, they found microbes trapped in giant crystals that may have been up to 50,000 years old. So that, was an incredible discovery, really surprised science. But then, just like a few weeks ago, scientists, and I'm sure there'll be maybe some debate on this, but they might have revived microbes that were 100 million years old. So it makes the 50,000 years old seem like a flash of a, blink of an eye. And they found them in deep sea sediments they were just kind of in a stasis state. And when they brought them into more habitable environments, the same cells that had existed 100 million years ago started reproducing. So it was like the same cell from before, and it hadn't really died, but it doesn't, lifespan doesn't really apply here in our usual sense of the word. And this is because some organisms can go into these stasis states and then persist for very long periods of time. But when they actually are thriving, their lifespan is much shorter. A cell's lifespan is not hundreds of millions of years old. It's just able to go into this stasis state. This fact blew my mind when I learned about it this last summer, which is that there are 1 million more viruses on planet Earth than there are stars in the universe. So coronavirus obviously is in the news. It's partly why we're having this online. There are 10 to the 30 viruses on planet Earth. And that's like just virus bits, not individual strains of viruses. But there are so many viruses because they're so small. And there are about 10 to the 24 stars in the whole universe. That's all the galaxies. So there's a million times more viruses here on planet Earth than stars in the universe. And so just to give you kind of these scales from the very small to the very big, and when we're thinking about life and the universe, I just think it's kind of an interesting thing. There's also this really interesting organism called Pando, which if you're interested, you can Google more. It's a tree cluster, and basically it has a single root system, and it just pops out clones of itself. And it's just been doing this for thousands of years. Some estimates put it at 80,000 years old. Some people think it's only 16,000 years old or maybe 1,000 years old. It's really hard to date because it just keeps, cloning itself and popping up new organisms. And it's like the single biggest organism on planet Earth. It's 6 million kilograms. And it's one organism with one root structure, and they all have the same DNA. So every single tree has the same DNA. It's not like daughter plants of the tree. It's all the same tree, just perpetuating itself. And each one, might live and die, but like collectively, it's like lived for these thousands of years. When we go to smaller things, there's these things called extremophiles. These are organisms that love all sorts of crazy environments. So they can exist in near boiling water, in Yellowstone, which is in Montana, where I'm actually from, or in these hydrothermal vents, deep in the ocean, where the temperatures and pressures are extremely high, and we wouldn't have expected life, and we're finding life there. We see life coming out, and living in Antarctica in really cold environments. And the life is living in the liquid brine layers between two ice crystals. So it's salty water, and that creates a liquid layer between the ice crystals, and that's where the microbes are living. So we're finding microbes everywhere we look, and microbes that are tolerant to radiation and temperature and salinity and desiccation, how wet or dry it is. It's pretty incredible. I don't know if you guys have heard of tardigrades or water bears, but they're the best. So they're a polyextremophile, and there's a little movie of one here. Keep in mind, like most extremophiles are single-celled microbes, but this is an animal. Like this is, has legs and claws and can be up to 1 millimeter in length. So it's very different, from most extremophiles because it's complex life, and it can withstand temperatures from well below freezing, even to near 0 Kelvin. So they've, to above boiling. They've put them outside of the International Space Station. I mean, they've done all sorts of crazy things to try to kill tardigrades. Tardigrades, what they do is they just enter this cryptobiosis state, and they're just able to survive. And then when conditions flourish, then they come alive and keep reproducing. So they can withstand pressures 6 times the deepest ocean trenches. So why? What is that for? It can withstand ionizing radiation 100 times what humans can withstand. The vacuum of outer space and those really cold temperatures and just vacuum. They can withstand without food or water for, I think it's now over 50 years that we've been trying to just starve them. Because again, they just enter this little biosis state, kind of like those microbes that were dug up from the deep sea. And so once they rehydrate and start to live again, their life cycle is much shorter, maybe a couple months. But during those states, they can just survive in this little cryptobiosis state for a long time. And all of these attributes of them fundamentally come down to their ability to dry out to 3% or less of their body water. And this ability to withstand dryness is what often yields the ability to withstand these other things, because it allows them to rapidly repair their DNA, and that then provides protection against radiation and whatnot, other things. So when we think about life is weird. You might have come across in chemistry that there's chiral molecules, so it's the same atoms make up the molecule, but they're mirror images of each other, so you can't put them directly over each other. And life uses these molecules in a very weird way. For example, life only uses left-handed amino acids and only right-handed sugars, by and large. Why is that? What if it used right-handed amino acids and left-handed sugars? And it's this handedness structure that causes the DNA helix to spiral. So we don't really know if this is necessary or not. This is an avenue of research within origins and within the origin of life. We might find life or even create life in the lab, which is opposite chirality. So that's like a whole area of research that I think is interesting. And when we're thinking about just like, well, what is life? It's really actually kind of hard to describe. So you can describe its characteristics, it grows, it replicates, it metabolizes, it evolves, it displays complex behavior. But non-life does all of those things too. So, crystals grow, computer programs replicate, and mules can't. Forest fires metabolize. Planetary systems evolve. Lightning and tornadoes display complex behavior. So that's not, those individual characteristics are not enough. And I like to think about this in terms of, well, what is water? Before we knew about atoms and the definition of water being H2O, 2 hydrogen atoms, 1 oxygen atom, any definition of water was insufficient. You could have tried to describe its characteristics like we did with life, saying water is wet and clear, but there are some oils that are also wet and clear. And with only one example of life in the universe, that's Earth life, are we really in the position yet to make a definition of life? We don't know if life's biochemistry is highly constrained, with that handedness. Can it be flipped? We just don't know. Or if there's many chemical solutions to life. So there's been many proposed definitions of life by various people. Life is the first encapsulating membrane. Life is a flow of energy, matter, and information. That's very physicsy. The most commonly used one is NASA's working definition, which says life is a self-sustained chemical system capable of undergoing Darwinian evolution. So this precludes then like synthetic, computer type AI life because it's a chemical system in this definition. But we don't really yet know until we find more life. I want to say like evolution is not goal-oriented. We tend to think of evolution going from simple to complex things or simple to intelligence. But that's not really how evolution works. It's just interested in, can you survive? Can you survive long enough to reproduce and have offspring? And as the planetary conditions change, what that means will also change. And I want to take a little brief interlude to talk about careers, because, as you think about what you want to do in the future, I've had so many things that I said I wouldn't do, and ended up doing things that I never thought I would do. So I wanted to briefly talk about that, and then you can ask me any questions, or we can talk about that at the end. My mentor, she said to me once, because I was really confused about what I needed a plan, I thought, of what to do. And she was like, you don't need a plan, Sarah, you need priorities. So finding out what are your priorities, and then the plan kind of follows from that. This is a post or like a graphic from the blog Wait But Why, which I would recommend. And it's a 90-year human life in weeks. So every little dot is a week of your life, and weeks go by pretty fast, actually. So you can see your whole 90-year life here on this sheet of paper and cross out each week. When you're 0 to 5, you're not really sentient yet at that point. And then when I was in high school and grade school and all of these things, I was planning to do anything except for physics. I hated physics. My dad was a physics professor. I never wanted to take physics as long as I lived. And then I got really interested in physics, so I majored in physics in college, but I hated astronomy because astronomy was very, we only know things by like a factor of 10. It seemed very imprecise and boring and looking at squiggly lines all day. And then I took a gap year off from my undergraduate before going to postgrad, and that's when I discovered astrobiology and started reading about it, learning about it, reading books, hearing lectures, and I was like, oh my God, this is the most interesting thing, and I want to go into it. And then from physics, the best way, I thought was to go through astronomy. So then I got my master's and PhD in astronomy. And then I moved to St. Andrews for 3 years, which was my first foray to the UK. And then, I should have increased this. So I've been in Oxford for 1.5 years. So imagine a few more lines there. But starting at Oxford. So James Webb is supposed to launch next year. Hopefully it will launch. And James Webb we'll talk about because that's like one of the first telescopes that can find life. And LUVOIR will actually have a much better chance if it launches. It's an even bigger telescope, and that really might be able to detect life. So Ray and I have talked about like when we'll find life. So we've made some bets. I've made bets with other people on when we'll find life on an exoplanet, on a planet orbiting another star. And so that's somewhere in my life in the future. So when you think about, your life, you're somewhere around here. What do you want to do with the rest of it? And this is, I think, what's great about the summer school is you get to hear a lot of different topics and think about all the various areas that exist. Because I remember when I was before I went to college and university, I didn't really know what existed fully out there. And so I think it's a really great opportunity to be in this school and hear all of different types of possible interests that might be there. So I also want to highlight a few things that were really important to me in my kind of exploring my career path. One is I came from a community college first, and that's if you're not, they don't really have them here in the UK, but it's basically like the lowest possible level of college that you can have. It's like they're not ranked or anything. They're just local colleges to help for rural students, for people who are first generation college students, etc. And so I had a lot of anxiety and self doubt on applying to Harvard for my PhD. So I eventually went to a state school, University of Calgary, basically for my undergrad. And because I had gone to a community college, because I was from like a rural community, I really didn't think that I would fit in or even get into a PhD program at Harvard. And I remember sitting on the application screen for like 90 minutes before actually hitting like submit, because I was just like, there's just no way. Why should I pay this stupid fee to like even apply? Like, I'm not going to get in. And then I applied anyway, and I got in, and I ended up doing my master's and PhD there. So don't say no to yourself is like the first thing that I would say. I think that often sometimes we take ourselves out of opportunities and, try to let someone else say no to you, but you make the application because you can't get it if you don't apply. And so I would encourage everyone to apply for things even when they don't think you're ready, or if you don't think you have a chance, because you just never know. And maybe you won't get into it. I apply to probably 10 to 15 times as many things as I actually get, So that's one thing. Then I gave a talk kind of along this topic, as well as just self-care. Your mental, physical well-being is actually the first step to you actually being successful, because we often don't prioritize that, thinking we'll do that once we've finished, studying for this final, etc. But the research has really showed it's the other way. Like, if you prioritize those things first, then you'll do better. And I have like a talk on YouTube, which you can see. It starts like 23 minutes into the video. You can just scroll ahead. And then if you're going to remember, the third thing is astrobiology is awesome. And I hope you all consider going into something that's related to astrobiology, because I think it's just one of the most cool fields in the world. All right, back to evolution, not being goal-oriented. Evolution produces really bizarre rituals. Like there's the bird of paradise mating ritual. You have like the human equivalent of our own dating norms can be rather ridiculous. And so when we think about evolution, and there's like evolution of life, but then there's just also, how do you go from a rock to the first cell? that's difficult, right? So there's like aliveness, non-life to life. And there's this quote that I like from George Whitesides that says it's a long way from slime to Mozart. And yeah, it took like 3.5 billion years. But it's a long way from HCN to slime. So just from a chemical molecule, hydrogen cyanide, to even the first simple cell is probably even a bigger jump in our current understanding. And we don't really understand this because like even the simplest cell is so complex. So we're starting to figure it out though. So that's what Origin's prebiotic chemistry research is all about. It's trying to find abiotic pathways to generate a metabolic cycle or to get RNA to form without any influence of life. So that's a whole fascinating area of research that I wish I had more time to talk about. But basically, is something kind of alive in a stepwise function? It gains some complexity and then it becomes a bit more alive? Or does it take a certain complexity and then it's all of a sudden alive? Or is it more a gradual process? We don't really know. But what we do know is the first living cell did not just appear fully formed with all of its chemical complexity. It must have originated in a series of stepwise emergent events, which we're starting to find out through prebiotic chemistry. So I just will leave that there and encourage you to consider that if you want to go into that area. As well, life and Earth have changed. So life on Earth has changed our planet has changed life, and so it's changed over the course of our history. So our Earth started off as like this molten ball of rock, not very habitable. I mean, chemicals can't form. So often when people think about, well, we just don't know about the limits of life. It's, what if it lives in the corona of the sun? It's like, yeah, but molecules have to be stable. There's some basic requirements, and I do feel like the stability of chemical molecules is one of them. Then you have the Archean Earth, early on with just kind of volcanic areas and tidal pools. We went through several snowball, fully glaciated phases. You had the Triassic period and the Jurassic period with the reign of the dinosaurs, all leading up to us. And so if you think about the history of life in units of 1 hour, this is roughly how it goes. So the formation of Earth was 4.6 billion years ago, or 0 minutes into the hour. We don't know when the origin of life started, but around 3.9 to 4.4 billion years ago, so that's 9 minutes into the hour. Our oldest signs of life are 3.5 to 3.8 billion years. Now, there's some people who are claiming that they found signs of life at 4.1 billion years. So we'll have to see if that turns out to be true. But either way, life seemed to have started very quickly, and we have the oldest signs of life dating back pretty far into Earth's history. It's hard to even have rocks from that time period because of plate tectonics. So it's incredible that we're seeing already signs of basically a global biosphere so quickly into Earth's history, or 14 minutes into the hour. The development of oxygenic photosynthesis took a bit longer because it's a more complex machinery, and that is 25 minutes into the hour, more or less. It could have been a little earlier. What I'm always surprised about is we don't really have multicellular life until 1 billion years ago. So that's 47 minutes into the hour. All of this life was just basically unicellular life. And then land plants, only 500 million years ago. That's 53 minutes into the hour. And then humans, this is anatomically modern humans, people that you'd recognize as your brothers, sisters, cousins, uncles, and aunties. They're only been around, we've been around 250,000 years, or the last second of this clock. So when we think about where to detect biosignatures, we can look on the solar system or we can look on exoplanets. And solar system has a lot of advantages because we can look for things on Mars or on Titan. We can actually send things there. Exoplanets were limited to a fuzzy, pale pixel, basically, which is a lot harder to disentangle. But a lot of what I do relates to exoplanets. And so what I'm interested in is, could we make future observations of a terrestrial planet system like Earth, Venus, and Mars and see the differences in those spectra and tease out which of those planets has life? And I'm interested particularly in this question of, are there biosignatures around other stars? So when I think about this problem, there's really two challenges. There's the technological challenge of just making a telescope big enough to do this, and then there's the interpretation hurdle. The technological hurdle is quite big. It's about technologically, if you were to think about detecting the atmosphere of a planet, or even just a planet from light years away, you're effectively trying to find a firefly in front of a spotlight. But imagine that that spotlight is in the US somewhere and we're observing from the UK. That's about how difficult it is, and maybe another factor of 100. So that's what we're trying to do with telescopes. And what's amazing is we actually are near that technological time point. So we're overcoming that technology hurdle in the 2020s. So there's a space-based telescope, James Webb Space Telescope. This will be the first telescope to actually be able to characterize the atmospheres of smaller rocky planets. It'll only be able to do a few of them, so not get a lot of data, but it'll be the first time we can do it. And then there'll be large ground-based observatories like the European Extremely Large Telescope that's also coming online in the 2020s. And so these will be the first telescopes, which I'm really excited about. And just in the interest of time, I'm going to skip some of this because we're going to need to move along. But basically, I think I'm really excited about this first opportunity. Is happening soon. It's happening within the next 10 years that we'll be able to get this first hint of can we see life on another planet. And on the 25-year horizon, we'll actually have telescopes big enough to do this in a statistically meaningful way. And so we can just look at the technological improvements over the ages of, say, Pluto. So this was our image of Pluto from 1930, which is basically how exoplanets look like to us right now. And then you have the Hubble Space Telescope in 1994, and then there was further improvements in 2003. This is a bit unfair because now I'm going to jump from a telescope that's in Earth orbit to a flyby, but now we can see Pluto with snow-capped mountains. You know, this is the image we have of Pluto in 2015. And I don't know if you're familiar with the Jupiter's Juno mission, but it's amazing. It's taken these high-resolution images of the planet Jupiter, and the images from it are just stunning. We can see even clouds on Jupiter. So there's ammonia clouds, there's water ice clouds, and we can actually image those now with this flyby mission. And these are falsely colored to highlight out different atmospheric structure, but like the physics happening is there. And it's just so beautiful, some of these images. So I recommend taking a little procrastination and diving down into Jupiter's Juno's images. But like when we think about the interpretation, the image will be difficult to interpret, especially as an exoplanet, because we're looking at this pale blue pixel. Right? There's really only one type of detection that would be unambiguous, and that would be a SETI, hello, here we are, sort of signal. Then we would know we found life. But everything else is going to be ambiguous. And so that's where my research fits in. So I do modeling atmospheres. I look at what wavelengths we should look at, what resolution of telescope, how big of a telescope do we need to have. And in particular, I also look at how does the star type, so stars are bigger and smaller, how does the star influence the atmosphere, the biosignatures, and what we'll see. We can think of, there's two ways of searching for life. There's life as we know it, and there's life as we don't know it. I'm not going to have time to go into that, so if we want to talk about it, and I want to leave some time for questions, the last 10 minutes, we can feel free to navigate that more. But in short, we could look for alternative biosignatures, but we just don't even know what those are because we don't have examples of life. That's like the short story. And so where I think alternative life, we should look in our own solar system because we can actually go and test those hypotheses, but it's impossible to do for an exoplanet. Then there's unique biosignatures on Earth, but those are often really kind of evolutionary quirks and they don't have a high abundance, so those are really hard to detect. And so then we're kind of left back with searching for Earth-like life, and that's things like oxygen, ozone, methane, and it's troubling because we would like to be more expansive in our definition. But on the other hand, water and CO2 are very abundant in the universe, and oxygenic photosynthesis relies on, light from the star, water, and CO2, and these are all very abundant. And carbon is more abundant than silicon; it forms more complex chemistry. water is polar and has all these weird properties, which makes it conducive to forming life. So this is why often, even though it's been decades of thinking about it, we still are going after some of these key molecules. And because of that, hopefully we'll be able to understand what we see. There's a lot of places to look first, because there's a lot of places like Earth out there. But the problem with these biosignatures is they all have a geological or photochemical false positive. So that means we could be fooled in what we're seeing. We could think we found life and have not found life. And so be skeptical of any first claim you read. These claims of life will be exciting, but there's always going to be a way that you could have fooled yourself. It's kind of, if you're going to take one sentence away. And in fact, there is a really exciting discovery that's going to be announced in September. And again, there's going to be a lot of debate. So I encourage you, when that is announced September 14th, pay attention, but remember that there are ways that we can fool ourselves. So it's worth being excited about, but also worth being skeptical. And yeah, so especially with exoplanets, we can be fooled. With solar system planets, we can actually go and try to figure it out. So those are kind of how I view those different areas. I think I'm going to end basically with this part of the talk, which is that the public, I think, thinks we're going to find Pandora, from Avatar, like a lush green, blue, tall people with tails and bioluminescence vegetation sort of world when we say we found a habitable planet. What theorists like myself claim we're going to find is this beautiful spectra, which is easy to interpret and has these nice, clear features. And the reality is it's going to be a pale blue pixel. There's going to be a lot of models that can fit the data. The data is going to be noisy. There's going to be high error bars on the data, and it's going to be hard to interpret. So I think I'm going to leave it at that and just see if there's anything else that is worth mentioning. I just want to go, I'll just end with these last two slides. One is that amazingly, we could maybe detect isotopes in exoplanets. So even detecting a molecule is hard, but for a quirk in science, actually at high resolution, isotopes could be easier to detect. I think this is fascinating. It just blew my mind that, an isotope is just a molecule with an extra neutron. It's just so exciting. So if you want to talk about that, we can. And then I'll leave it on this slide, which is, science is a way of trying not to fool yourself. And the first principle is you must not fool yourself, and you're the easiest person to fool. So I think you'll see this. I think you'll see it soon, that we scientists, we want to believe that we'll find life. And so we're predisposed to that. And sometimes we can get overly excited. But science as a discipline is self-correcting, because other people will be like, well, what about this? What about this? Did you think of this? And over time, it moves forward in a slow fashion. Which is what I'm really excited about, is, science as a discipline rather than individual discoveries, because those are always going to take time to figure out. All right. Thank you so much. And we'll pause there for questions.